This is an introductory lecture to provide some information that'll be helpful for doing the tree biology lab uh, wood ID. I sometimes call this the lumber biology lab. An important concept to grasp when thinking about wood and wood ID is that all of these cell types that we've talked about in class exist within a three-dimensional cylinder. And that sometimes makes it complicated to think about how these cells relate to each other and what they look like, for instance, if you slice a stem different ways. I really like this diagram because it demonstrates three main planes that a tree stem, a cylinder, can be sliced in, and it shows how the broad morphological features of a stem show up. So one of the main sections that we use for identifying wood and for looking at wood is the cross section. So if you took, a cylinder like this and cut it in this direction and then looked at it this way, that's the cross section. We're used to seeing tree stems in this section. That's how growth rings show up. So this is a pine stem. So these dark bands are the late wood bands, and the light bands are the early wood bands. And we're sort of used to seeing wood in that section. But another common section that we might see from a stem is a radial section. And that would be like taking a pie cut out of a stem and cutting that pie along a radius and then looking at that radius from the side. So this plane is a radial plane. And you can see that when we cut a radial plane, the wood looks like this. We're still seeing early wood and late wood bands, but they look different. So those dark lines that we're seeing are still the late wood bands, and those light lines are the early wood bands. They're just exhibited differently because of the nature or the plane that we cut them in. So we're viewing all those late wood and early wood tracheids basically from the side instead of from the end. And then the third plane section that we might see is a tangential section. That would be if we took the stem and cut it along any plane except right through the middle. If we took a slice, that would be a tangential section. And you can see, again, we're seeing light areas, which are the early wood, dark areas, which are the late wood, but it looks a lot different. That's because, for instance, in this tangential section, as that section was being made, it never is going to follow perfectly along an early wood or late wood band because it's not perfectly straight up and down, and the tree doesn't grow perfectly in that plane. You're going to see late wood coming in some parts of that section and early wood coming in other parts of that section. So that gives sort of this random look. These, patterns that we see on these different faces are what we would refer to as wood grain. And so wood grain is basically the visual patterns that we see in the wood that is the interaction of the anatomy of the tree with the plane of cutting that was made. So these planes are sometimes, we use this terminology in biology when we're making different sections, but it's also pertinent to, for instance, lumber and wood grain in this context. So the most common way of cutting lumber, so solid wood, from a log is to plane saw it. Plane sawing is just when you take the log, lay it on a cradle, and then take the blade, this is a bandsaw blade, and just sequentially make cuts through that stem, and each of those cuts produces a board. And all of those cuts, except the one right through the middle, the one right through the middle will be a radial cut, but all the other cuts are tangential cuts. And that's the typical way of producing lumber, is plane sawing. This is a portable sawmill, but industrial sawmills usually use bandsaw blades as well, and usually those boards are cut by plane sawing. But in some cases, people may choose to quarter saw wood. And this is an example of a quarter sawing setup being done. So the log is literally quartered and then rolled on its side. Those quarters are rolled on their side so that each cut is on an approximately radial plane. The reason this is done is that for some woods, especially like oaks, a quarter sawing or a radial plane will produce special grain features. So this diagram shows, in this case, an oak log and how the cuts might happen with a quarter sawing setup. So this is white oak that's been quarter sawn, and this is white oak that's been plane sawn. And you can see the grain pattern is different in that plane sawing versus the quarter sawing setup. And that's because this is a radial section. And in this case, these dark lines we're seeing are actually the early wood in that white ring porous white oak, and the light bands are the late wood in that ring porous oak. And the same here, except the pattern is different because this is a plane sawn or tangential section. This is a quarter sawn or radial section. The main reason this is done is because in oaks, that radial section or quarter sawn plane will produce these ray flecks. It will reveal the rays. And that's desirable for some styles of furniture. So this is finished quarter sawn white oak, and you can see these beautiful reveals of the rays that are revealed when that wood is quarter sawn. So when oak is sawn for furniture, it sometimes is quarter sawn. And then finally, when we, for instance, produce plywood, that plywood has a pattern on its surface that looks like this, that looks like tangential section, and that's because plywood is produced by making veneer. Plywood is basically large boards made by taking thin pieces of veneer that are peeled off of logs and placing them at 90-degree angles to each other and gluing them together to make a plywood board. So those pieces of veneer are peeled almost like paper towels off a paper towel roll. But in this case, there's a knife that's placed up against a spinning log, and the pieces of veneer are peeled off of the log. So I'll show you that. So that log is spinning and being pushed up against a knife that's producing these sheets of veneer that'll be turned into plywood. So that's why if you look at the surface of a piece of plywood, it'll have its surface that looks like this, because essentially that log was peeled off like a paper towel roll. So again, I'm trying to get you to visualize the cells that we've learned about and the patterns they make in wood and how that translates to some of the products we see in our everyday lives. In this lab, I'm going to have you go through some very simple and fundamental steps in identifying wood to species or to genus. The actual process of identifying wood can be quite complex and involved. And I really encourage you, if you're interested in going deeper into this topic, to acquire this book, Identifying Wood by Bruce Hoadley. It's a really nice book, has some beautiful illustrations. I use a lot of them in this lab, but there are lots of keys and details in there on how to really dive into Wood ID in great detail, much more detail than we'll cover in this lab. So I encourage you to get this book. Sometimes people make fun of it, identifying wood. Yep, it's wood, which really isn't fair because this is a really neat topic. So you can find this on Amazon or other booksellers. It's really not very expensive. So I encourage you to do that if you'd like to have that on your shelf. But I'm just going to walk through some of the very simple things you can do to start identifying a piece of wood that you might have at hand. So the first thing to do is to determine if there's something really distinctive about the piece of wood you have. There are some woods, like if you have a piece of heartwood that has a very distinctive color, like black walnut, or a very distinctive color and smell, like eastern red cedar. If it's very heavy or very light, sometimes that by itself might allow you to really narrow down what that wood is, and maybe stop there. Eastern red cedar is so distinctive, you probably don't have to do additional steps. But that's sort of your starting point. Is there something really distinctive about this piece of wood that can help me understand what it is? That's usually not the case, and if that's not the case, you're going to have to do some additional steps. So for our purposes, the next step is to determine whether that wood has vessels or not. Now, you determine that by taking your piece of wood, determining where the cross section would be, and creating a nice clean face on that cross section, either using a sharp pocket knife or a single-edged razor blade. And then you can use usually a 10x magnifying glass to look at that cross section. And in that cross section, you should be able to determine whether the wood has vessels or not. So these two examples, these large holes in this cross section here, these are vessels. Whereas here, we can see these with vessels will have sort of large openings that are within a matrix of fibers. Okay, whereas here in this example, these are just all tracheids all jammed together. We don't see fibers. So this distinction, whether they're vessels or not, allows us to determine, okay, this is an angiosperm if there are vessels, and if there are not vessels, it's a gymnosperm. So that's sort of our first big division. If it's a gymnosperm, well, we got problems because gymnosperms are difficult. This is a binary or a dichotomous key from Hoadley that sort of allows us to do some initial groupings of gymnosperms, or what Hoadley is calling softwoods. And we'll sort of do a couple of these steps in this lab. So if we've determined that it doesn't have vessel elements and that it's a gymnosperm, we can say, okay, does it have resin canals? If it has resin canals are sort of rough openings. I think they look sort of like rough openings in the cross section that are larger than tracheids, but they're not sort of distributed across the growth ring like vessels would be. So in these two cases, we have resin canals. In this case, the resin canals are larger, and they're often located in the late wood, but not always. If there are large resin canals, then it's likely a pine, a pinus. If there are smaller resin canals, and of course this is a relative thing, but if the resin canals are smaller, then it's likely a Douglas fir, a spruce, or a larch. If there are no resin canals present, then it's another gymnosperm. So that could be a hemlock, a true fir, a cedar, bald cypress, redwood, or yew. So that's a lot of possibilities. In the case of those other, we often then would need to go to microscopic characteristics. And we're not going to go into that in this class, but as an example, this is an image from Hoadley where he shows how to use microscopic sections or thin sections taken from wood, and you search around on a microscope and you can find bordered pits. And the differing structure of bordered pits can sometimes help you to separate out, for instance, in this case, spruces from larches. Okay. There are many other characteristics. So for instance, Douglas fir has helical thickenings in the tracheid secondary walls. And those are some of the kinds of things that unfortunately you often have to get into to separate some of the gymnosperm species. Again, we're not going to go into that in this class. The next step, so that's sort of gymnosperms. For angiosperms, if you've determined the thing has vessels, the next step is to say, does it have rings or not? You know, rings, we're used to seeing rings. This is what rings look like. If the thing has rings, then it's a dicot angiosperm. If instead the vascular system is distributed across the stem in vascular bundles, then that tells us it's a monocot. In our case, that would, if it's a tree, it's a palm. Okay, so palms are really the only monocot tree that we talk about in this class. So if we have vascular bundles like this, it's a palm. Anything else, if it's got rings, it's a dicot angiosperm. Dicot angiosperms are diverse, but there are characteristics we can use to determine dicot angiosperms. So if you've then determined the thing has vessels and it's got rings, then you have a dicot angiosperm. So for our purposes, then the next step would be to determine how the vessels are distributed across the growth ring. And you remember from lecture, we talked about two main types of distribution, ring porous and diffuse porous, where ring porous is a situation where very large vessels are produced early in the growing season. So we have a few rows of really large vessels, and then there's a rapid and abrupt transition to much smaller vessels that make up the late wood. That's ring porous. Diffuse porous is sort of the opposite, where we have relatively uniform size distribution of vessels all across the growth ring. So there really isn't a gradation in vessel size across the growth ring, which means we really can't determine where early wood and late wood is. I didn't talk about it a lot in lecture, but for the purposes of wood ID, there also are intermediate, there's an intermediate classification called semi-ring porous or semi-diffuse porous, where there's a gradual gradation of vessel size from large in the early wood to small in the late wood, and it's a gradual gradation. So different species tend to produce vessel distributions that are ring porous, semi-ring porous, or diffuse porous. And the key will tell you which one is likely. So that distribution of vessel size can be a really important determining characteristic. So just to back up, just want to make sure we're clear on what we're looking at in these cross sections. So this is a ring porous cross section here, and The number 1 here is an entire growth ring. So this is 1 year's worth of growth with these large vessels in the early wood and smaller vessels in the late wood. So this number 3 is early wood and number 2 is late wood. So this is the correct, answer for identifying these parts. Another thing that we can use to determine down to genus or species sometimes is that there may be other patterns of vessel arrangement. So in this case, these are all ring porous arrangements. You can see all of these examples have very large vessels in the early wood and then an abrupt transition to small vessels in the late wood. But in this case, in all of these examples, the late wood vessels are arranged in sort of these wavy patterns. The late wood vessels are all arranged in these sort of rows that are wavy. When that is the case, that's a sure indication that this is elms or hackberries. So, Hoadley calls these ulmiform arrangements. These wavy patterns are ulmiform patterns, where ulmus is the elm genus. So that's pretty handy. So in this case, that's an example of a particular patterning of vessels that may give us additional clues as to what species that may be. And then another thing that might show up or that we might look for is really prominent rays. So this is a cross section from an oak. Oaks are ring porous. You can see this has that classic ring porous arrangement, but in this case, there are very prominent rays in oak. And those prominent rays can also be a good thing that helps us to separate out one species from another. So for the purposes of this lab, I've created a really simple dichotomous key for our 5 unknowns that you can work for. So, we'll start by looking at the polished cross section on those samples and saying, are there growth rings present? Well, no, it has vascular bundles. In that case, it's a monocot, which means it's a palm, in this case, sable palm or sable palmetto. If it does have growth rings, that means it's either a dicot angiosperm or a gymnosperm. So the next thing would be to say, okay, does it have vessel elements? If it does have vessel elements, then it's a dicot angiosperm. If it doesn't have vessel elements, if it just has tracheids, then it's a conifer or a gymnosperm. Okay? If you've determined that it's a dicot angiosperm, look at how those vessels are arranged. Are they diffuse porous or ring porous? Well, in our unknowns, we have one diffuse porous species, and that's yellow poplar. If it's ring porous, we have one ring porous unknown, and that's red oak. If you've determined that it's a conifer or a gymnosperm, you can look and see if it has resin canals. If it does have resin canals, then we can get it to genus, it's pine, Pinus. If it doesn't have resin canals, then our unknown is either a fir or a hemlock. Often firs and hemlocks are grouped together because they're very difficult to separate just based on macroscopic features that we would see in 10X. We would have to go to the microscopic characters to separate them further. So for the purposes of this lab, this dichotomous key should allow you to key out the 5 unknowns to species. So these are the unknowns. So this is number 1, number 2, number 3, number 4, and number 5. So you should be able to use these cross sections to key out to species those 5 unknowns. In addition, in your lab report, I'm going to need you to create a figure, at least one figure, for each of those unknowns and label the different cell types that you can see on those cross sections. In some cases, you won't have a particular cell type, but I want you to identify the cell types that you can see in your lab report.